Dear students, you are welcome back to STR Shivarajan's teaching room. The last episode we had discussed about the stamens and the structure of microsporangium. In this episode we are going to discuss about how the pollen grains or the microspores are formed. So the formation of microspores or pollen grains from the microspore mother cell or pollen mother cell after meiosis is called a microsporogenesis. So we are going to discuss about microsporogenesis. What is microsporogenesis? The formation of pollen grains or microspores from the pollen mother cells or microspore mother cells after meiosis is called a microsporogenesis. See, look here. The microsporangium has got four layers: epidermis, endothecium, the outermost layer epidermis, the second layer endothecium, the middle layer, the third layer, the middle layer, and fourth the tapetum. Inside the tapetum, see, there is a gap, there is a cavity. This cavity is the microsporangium. Inside the microsporangium, there is a tissue. See, I have drawn some tissues in this cavity now, and these are the cells in the microsporangium. The cells are of the same type. All the cells in the microsporangium are the same type, so we can say that this is a homogeneous tissue. Homogeneous tissue. Homogeneous means the same type of. So all the cells in the microsporangium are of the same type. So there is a homogeneous tissue inside the microsporangium, and this homogeneous tissue inside the microsporangium is called a sporogenous tissue. Sporogenous tissue. So there is a tissue inside the microsporangium. The tissue is made up of same type of cells. So it is a homogeneous tissue, and the tissue is called a sporogenous tissue. It is called sporogenous tissue because it is from this tissue that the spores are produced. And the cells in the sporogenous tissue are diploid. Okay, now what happens? Differentiation takes place in the tissues in the sporogenous tissue. Differentiation means change. Differentiation takes place in the sporogenous tissue, and they become spore mother cells. They become pollen grain mother cells. Pollen grain mother cells. So the cells in the sporogenous tissue undergoes differentiation, and they become microspore mother cell or spore or pollen mother cell. So the tissue inside the microsporangium is made up of the same type of cells, so it's a homogeneous tissue, and this homogeneous tissue is called a sporogenous tissue, and the cells in the sporogenous tissue undergoes differentiation, and they become microspore mother cell or pollen mother cell. And each microspore mother cell or pollen mother cell is diploid. It undergoes meiosis. So from a pollen mother cell, four cells are produced. Four haploid cells are produced. As a result of meiosis, from a single pollen mother cell, four haploid cells are produced, and all the four haploid cells remain together. So it is called a tetrad. Tetrad. So it's called a microspore tetrad or pollen tetrad. Microspore tetrad. And on maturity, these four cells get separated and they become independent, and they develop into pollen grains. And each pollen grain contains the male gametes that we will see later. So what is the form? How the pollen grains are formed? The microsporangium has a homogeneous tissue called sporogenous tissue, and the sporogenous tissue undergoes differentiation and they become microspore mother cells or pollen mother cells. The microspore mother cells or pollen mother cells are diploid. They undergo meiosis and 4 haploid cells are produced. All the 4 haploid cells remain together. Then it is called a microspore tetrad. Tetrad means a group of 4 cells staying together. And the microspore tetrad on maturity, what happens on maturity? The 4 cells get separated and they develop into pollen grains. This is microsporogenesis, and on maturity of the pollen grains, the microsporangium breaks. It breaks at the point of, at this point of the space where both the theca join here. This is a theca, this is another theca. So it is at this point, it breaks and the pollen grains come out. So what is microsporogenesis? Microsporogenesis is the process of formation of microspores or pollen grains from the microspore mother cell or pollen mother cell after meiosis. Once again, if you go through this, see, the microsporangium has a homogeneous tissue called sporogenous tissue. The cells in the sporogenous tissue undergo differentiation and they become microspore mother cells or pollen mother cells. These cells are diploid. These cells undergo meiosis and 4 haploid cells are produced. All the 4 haploid cells remain together. Then it is called microspore tetrad. On maturity, these 4 cells get separated and they become pollen grains or develop into pollen grains. This total process is called microsporogenesis. Okay. So, what is the structure of a pollen grain? What is the structure of a pollen grain? A pollen grain has, see, pollen grain. A pollen grain has got 2 coverings. This outer covering is called the exine and this inner covering is called the intine. The exine has got some openings here and there. Here is an opening and here is an opening. Okay, or here may be an opening. So the exine has got openings here and there. They are called germ pore. This is the germ pore. And it contains 2 cells. One big cell called the vegetative cell, and the small one is called a generative cell. Okay, this is the vegetative cell and this is the generative cell. The vegetative cell is bigger one and the generative cell is a smaller one. So the pollen grain has got 2 cells. 60% of the pollen grains are in this 2-celled stage when they are released from the anther. 60% of the pollen grains are in the 2-celled stage when they are released from the anther. Okay, the other 40%, remaining 40%, they know are in the 3-celled stage, and after releasing from the anther, the generative cell divides mitotically into 2 and becomes a 3-celled stage, and these 2 cells are the male gametes. So, and the exine is made up of a chemical or substance called sporopollenin. Sporopollenin. The exine is made up of a substance called sporopollenin, and it is such a substance that it cannot be degraded or it cannot be destroyed by any non-chemical substance till now. So any non-alkali or any non-acid or any non-chemical substance cannot destroy sporopollenin. So it protects the pollen grains for a longer time. So this is the structure of a pollen grain. So today in this episode we have discussed about the formation of pollen grain, that's microsporogenesis, and again we had seen the structure of a pollen grain. In the next episode we will see the other details of the pollen grain, the viability of pollen grain, its importance, and things like that. So we are winding up this episode at this point. So in this episode we have discussed about microsporogenesis and the structure of pollen grains. Thank you for listening to me. And please note to subscribe, the viewers may please note to subscribe this channel and press the bell button so that when I upload a new episode, you will get a information or notification so that you can view the coming on episodes. Thank you.